As the genetic counselor for the autism research study, my main role is to provide feedback about genetic test results to families. So it's a pleasure to be here to share results from the research study to a broader audience. So the title of my talk today is Genes, Variants, Results, and Impact. These are the 4 categories that I hope to discuss with you today. So the first thing I'm going to talk about are genes, giving you a review of genetic concepts. Then I'll talk about variants, types of changes, and the genetic tests that can identify them. Then I'll talk about the results, what we know so far from research, and then the impact, what it means for families. And so hopefully, in addition to sharing results with you all, I hope to give you some foundations with regards to the genetic terminology that you may hear throughout the day. So to begin, with neurodevelopmental disorders, including autism, there's no one gene that causes autism or OCD or intellectual disability or ADHD. It's considered a multifactorial condition. So what that means is that there are many factors that play a role, including environmental factors and genetic factors. And you can see there are different impacts or strengths of these factors. And the way I like to describe multifactorial conditions is using a cup analogy. So if you imagine this cup as an individual, and in order to develop the neurodevelopmental condition, you need to fill this cup with risk factors. So for each individual, they're going to have a unique combination of risk factors. And it's not until the cup is filled that one would develop that condition. Okay. So that's what's considered multifactorial inheritance. So when I talk about risk factors, what am I talking about? So today I'm going to focus on the genetic risk factors. And in order to understand that, I'm going to start first with the human body. So our bodies are made up of trillions and trillions of cells. We have skin cells, hair cells, brain cells, heart cells. And in nearly all the cells of our body, it's the genetic information that tells our bodies how to grow and how to function properly. And these instructions are organized into structures that we call chromosomes. So if you unwind the chromosome, you'll see the genetic sequence. There are sections on the genetic sequence that are called genes. So you've probably heard the term genes before, but what is a gene? A gene is important because it is the instructions that code for a protein. And you can think of proteins as workers in the body. They have a specific job. So we have proteins that work in our heart, we have proteins that work in our brains. If you want to learn more about basic science and genetics, these are some helpful websites. They have great videos on them that go through animations. So a genetic variant, a genetic change, a genetic risk factor, what does that mean? So we think of this as a change to the instructions that code for a protein. It disrupts that instruction so that either the protein is made incorrectly and therefore can't do what it's supposed to do in the body, or the instructions are disrupted in a way that the protein's not made at all, so it's not working at all in the body. So that's what I'm talking about when I'm talking about a genetic variant. So in the next section, I'll talk about the different types of variants and the tests that we use to detect them. So for most of the families in here today, to enroll in the research study, you most likely had to give a blood sample, or your child did. That blood sample is a source of blood cells, and it's in those blood cells that we can see the genetic information. So these are the chromosomes. Okay, so if we actually look at a blood cell under the microscope, we magnify it, you can actually see that there are 46 chromosomes. And they come in pairs, so 23 pairs of chromosomes. One pair of chromosome comes from our mother, the other pair of chromosome comes from our father. You can think of this as books on a shelf. So if we had a bookshelf, you should see 46 books, 2 copies of every book. This type of technology of looking at the chromosomes under a microscope in a magnified view and organized is called karyotyping. Okay, and karyotyping can identify large changes in the structure of the chromosomes. So going to that book analogy, if there was an extra chromosome, so for example, in this, what's illustrated there is an extra chromosome number 21, which you can think of as having an extra copy of book number 21. You would be able to see that using this karyotyping technology. And this is the case, the cause of Down syndrome in the majority of cases. You can also similarly see changes where there's missing chromosomes or missing parts of a chromosome. So again, on chromosome 5, there's a little section that's missing. You would also be able to see this using karyotyping. Now, imagine, though, that the structure is not changed. But instead, I take a book out, I open it up, and I either rip out a couple of pages or insert a couple of pages. I close that book, put it back on that shelf. Just looking at the bookshelf, you wouldn't be able to see a difference. So the next type of genetic testing that we use is called microarray analysis. And what this technology is able to do is look for missing, what we call deletions, or extra, what we call duplication, pieces of genetic information. Collectively, deletions and duplications, they'll also sometimes be referred to as copy number variants, or CNVs. So you can think of microarray technology as going through all of the books and counting the number of pages to make sure that they're all there. Going one step further, now imagine all the pages are normal, are fine, but I go into a paragraph, and instead of one letter, I switch it out for another letter. And you could see that switching out the letter could change the instructions of that sentence. And so the newest form of genetic testing that we have is called whole exome sequencing or whole genome sequencing. Sometimes it's also referred to as next generation sequencing. And what this technology is able to do is looks for spelling mistakes in the genetic information. So you can think of it as a large spell check. So that's the overview of the technology, the different types of genetic variants that we can see. Large structural variants, deletions and duplications, and spelling mistakes. Now, so that's what we're looking for when we're doing these research studies. Are there differences in the genetic instructions of individuals with a neurodevelopmental condition compared to individuals who don't have a diagnosis? Keep in mind, though, all of us, each and every one of us has changes to our genetic instructions. Some of these changes, we know, contribute to health and developmental difficulties. Some of these changes, we know, don't have a direct impact on our health and development, and they're considered normal variations that make us stiff and are seen in the general population. Then there are variants where we're not quite sure whether they contribute to health conditions or not. So at this point in time, they're described as uncertain. We see them in some individuals who have the condition. We also see it in individuals who don't have the condition. So we're kind of limited in terms of making a call one way or the other. And Esther from Autism Speaks is going to talk more in depth about the Missing Project, but just to highlight here that this new project, which most of our research team, actually most of the families in here will soon be a part of, will help accelerate this analysis stage of determining the significance of genetic variants one way or the other. Okay, so the next section I'm going to talk about is results. So what do we know so far? From our research, we've learned that there are hundreds of genes that play a role in causing autism spectrum disorder. So there's no one gene that causes autism. And you can see from this illustration that the genes are across the entire genome, so they're on different chromosomes. There's not just one chromosome that's affected. I've listed here 3 examples of genes that have been associated with autism spectrum disorder, Nerexin-1, Shank2, and PATCH-D1. These genes, and most of the genes we've identified, play a role in brain development or function somehow. So we've identified genes that encode proteins that are important for making connections in the brain. We've identified genes that are important for the actual growth of brain cells and brain cells or neuron signaling. And Evdokia already mentioned this as well, but we've identified variants in the same gene having implications for many or different neurodevelopmental disorders. So a variant in one gene has been seen in intellectual disability. It has also, the same gene has been implicated in autism and in psychiatric disorders, as well as seizures. So there, in addition to the phenotype overlap, so the symptoms and the between these conditions, there's also a genetic overlap. So it's important to study all of these conditions together. More recently, we have learned that within the same family, siblings with autism, so two individuals with autism, will have variants in different genes. So even though they're from the same family, the contributing factor to the autism could be different. So in this next section, I'll talk about, so what does this mean? So I'm talking about variants and identifying changes, but to families, how does that benefit you? So if we identify a variant that we think is contributing to the autism spectrum disorder or the related disorders in the family, this might help direct medical management. So for example, if we know that a certain group of individuals with the same genetic contributing factor also have seizures, heart disease, or are at increased risk for obesity, then we can make recommendations in order to get screening for these associated conditions so that it can direct the care for that child. Learning that there's a genetic variant can also help us as genetic counselors refine the recurrence risk for the families. So what is the chances of having another child with autism? So in the past, when there was no genetic, if we don't know the underlying genetic contribution, then we have to rely on empirical data, which is where they study a group of families where one child is affected with autism, and they go on and count how many of the subsequent children do they go on to have autism. And the numbers the studies have produced are between 10 to 20% of couples who have one child with autism will go on to have a second child with autism. And that's the limited information we have, so not a direct number that we can give to the family. That's different if we know the genetic underlying contributing factor or cause. So for example, if the autism is a part of the underlying Fragile X syndrome, then the inheritance pattern is X-linked inheritance, where the chances is as high as 50%. So I just also wanted to mention here, for the genetic testing that I talked about is for a child with autism, or an individual with autism. It's not for prenatal testing. So if you're a couple and you're thinking of having a child, there is no genetic testing for autism at this time. And so it's only when there is an underlying diagnosis that we've identified where we can refine that risk and talk about options in terms of prenatal testing. And lastly, identifying a genetic variant is also helpful for the family in order to connect them with the community and with support groups. So if families with the same genetic findings come together, they can share their stories, their experiences, and give advice. And lastly, what does this mean for society? So I've talked about the contributing factor, the impact for families, direct families, where I will meet with them and give them the results and talk about what it means and next steps. But for some families, the majority of families, all the results come back and they're normal or they're uncertain. And in this case, it's not though. Collectively, all these families do contribute to a benefit to society, which is that it helps clarify our understanding of the genetic contributions to neurodevelopmental conditions, which helps direct our initiatives to better characterize subtypes based on genetics so that, one, we can give families information about what to expect, and two, we can provide physicians with recommendations in terms of how to care for these children. This better understanding of genetic contributions also helps give insight into earlier detection approaches to identify individuals at risk in order to start earlier intervention. And then lastly as well, it also helps, it's the foundation for the development of targeted medications. So that's a lot quicker than I had anticipated. So I'm happy to take any questions. Timing. Right, so we know that our genetics is when we're born, it's with us, but the other factors, the environmental factors, can come at any time during development or afterwards. And there's what's not illustrated in this, it's a little bit more complex, is the interaction between environment and genetics, where the genetics might be the same, but certain environmental triggers can affect how that gene is expressed. So that could be at play with regards to what we're seeing with different timing.
Hi, we have identical twin boys that were diagnosed on the spectrum several years ago. They're 11 now. They were 4 years old when they were part of the study. We did get some information about the genetics breakdown. There wasn't too much information to give at the time. In the 7 years past and things that were learned, is there any way to look at their, and I guess anybody else's that are here, look at those results and see how the significance may have changed over the last 7 years?
Yes, so that's perfect, and that I didn't cover that in much detail either, but the genetic information stays the same. Our genes are our genes and the sequence is the sequence, but what you alluded to is the significance. So right now, that's the hardest piece, determining if it's playing a role or not playing a role. We know that there's a change there, but what does that mean? We're uncertain about. So in terms of the reanalysis, that's constantly being done. So at this point in time, we have a list of the variants that we think are significant. As we collect more and more sequences and compile the evidence, it'll push it one way or the other. And so if it pushes it to the significant factor, then we might be calling you back and saying, you know, that variant that we didn't know much about 5 years ago, we now know it seems to be playing a role, and we'd like to do ABC. So that is currently being done.
One of the slides that you had showed that if a family has a child with an ASD diagnosis, there's a 10 to 20% chance of an additional diagnosis in another child.
Yes.
Is there any information with regards to of the children? If someone has an ASD diagnosis, is there a percentage or any information of if they have a child then? What percentage of someone with an ASD diagnosis, if their children then would?
Yes, so there hasn't been studies yet. We're looking at individuals with autism and what are their chances of having a child. So we would expect it to be higher, but the data, we just haven't done studies. So those data, the 10 to 20%, is based on empirical studies, meaning that you just take a family and you count how many go on to have a second child. That hasn't been done where they take a family where the parent has autism and count. So we're limited in terms of that right now. We've had questions about even an unaffected child, what is their chances, so a second degree, and we don't have the data right now.
You talked about when you have a specific deletion, potentially that there could be you could link them up with other people who have that. How does that happen if you do have a specific deletion? You actually mentioned on the slide the actual one that I have. Yeah. So I'm just wondering how we would get connected to that, because it's new research, I know, specifically on this one on the 16th that is sort of coming to light. How would we get connected with other families?
Yeah, so when I see a family, what I'll often do is do the research first in terms of what is out there with regards to community support and from the research perspective as well, because there are researchers looking specifically only at that one deletion or duplication, and I'll provide that to the family. But if you want to go on your own and check, a great website is the Unique. This one here. The Unique website, which is Understanding Chromosome Disorders That Are Rare, and they tend to put together pamphlets that are parent-friendly, and within that you can sign up for their community, and then they'll connect you with other families. The other way is through Facebook. A lot of families will put groups together where they have that one deletion or duplication.
What if it's not an actual disorder? It's not listed as a disorder as of yet, but I know
Right.
that there's links towards, that you've looked at links towards certain things, but you're not exactly sure, but you know it's
Yeah.
certainly possibly something, like
Right.
the 16th chromosome and obesity and
Right.
autism. 'Cause I
So
know-
so the rarer ones you're saying, where there's
Yeah.
nothing already established? Yeah, so
Rare.
from our research lab, we do have a list, so we know how many families there are.
I assume that's confidential though, unless they want to.
Yeah, so usually what will happen is we connect with the other family and say, are you interested in meeting with another family? And we'll do it that way.
So I would have to speak to my genetic counselor?
Yes.
Okay,
Okay,
thanks.
no problem.
Hello. I wanted to ask if environmental factors are gender neutral as far as affecting development of autism.
Sorry, I heard parts of your question. Are general neutral environmental
I
factors?
wanted to ask if environmental factors are gender neutral as far as affecting the development of autism.
Is that the question?
It's
Is
a
it?
question,
Okay.
yes.
So I would say yes and no. So I guess it depends. There are certain genes that have different expressions in males and females, especially the ones on the X and the Y. So depending on environmental triggers and what its gene is impacting, it could be different expressions.
Well, the reason I ask is that to me, the ratio of boys and girls that diagnosed with autism suggests strongly that it's genetical, but wouldn't if environmental factors are at play, wouldn't that move the ratio towards being more equal?
Yes, so the male to female ratio of 4:1, part of it can be explained with genetics. So there are some conditions and some genes that are impacted that are on the X or the Y. So they're going to be different for males and females, but it's not the full picture. So you alluded to the fact that environmental factors might be at play. And there's also, there could be some sort of protective factor that is in females and not in males. So we're not quite sure. But in terms of the syndromes that, like fragile, or when you look at the autism that's caused by a genetic factor, the ratio of 4:1, it's more closer to 1:1.
Thanks.
When I first started talking about autism coming to these conferences, I think there was a question as to if genetics played a role in every single diagnosis. I know you haven't identified how genetics affects every child with autism today. Would you say there's a working hypothesis that there is a contribute of genetics in every child with plus environment? You just haven't yet identified it?
Yes. And going back to that multifactorial inheritance, those genetic factors could be the small ones, right? Not where it's easier to identify the big genetic risk factors, where having it, you get autism, or very close to, with a few other triggers, you get autism. It's the other ones, where having one may not present unless you have other things. Those are a little bit harder to unravel. But I believe that genetics is a part of all cases.
One other question. Do you know if there's any work going on, or how much work is going on with respect to siblings and other things that may occur with them, for example, ADHD, LD, things that are non-autistic?
In terms of assessing them?
In terms of the correlation, in terms of people researching that?
So a part of our study actually is looking at that, where we're bringing back families where there is a genetic change, and we're looking at other family members, so parents and all the siblings. And we're seeing, is there a pattern between those who have the genetic change and those who don't, even within the same family? And then we're also similarly bringing back families who don't have that change and doing the exact same thing, so that even though that genetic change may be presenting in one child with autism, but it might be presenting as OCD that's more subtle, or ADHD in other family members.
Hi. You mentioned that children with ASD have difficulty with connections in the brain and brain cell growth and neuron signaling. And so now I'm wondering, I've read some articles and so on the plasticity of the brain and the ability to reroute or rewire and all this thing of patients who have gone through this. And so I guess my question is, genetics are genetics, but it's positive to think about how we can reroute. Now, is there any connection then? Like if something's genetic, can we still reroute? Can we still rewire? And is this plasticity of the brain, is there some connection, I guess I'm asking, between that? Or is genetics genetics? And I guess that's my question.
So I think there's two questions there, and Evtiki, I can talk to about the networking piece of it, but in terms of the genetics, we're not, so by identifying a variant in a gene, the approach is not to fix that gene and put it back to correct the network. That hasn't been the approach. It won't work. But does that mean, though, that just because the variant's there, we can't do anything about the network? I don't think that's the case, and Evtiki can talk about that piece of it, but they're two distinct things between genetics and then the networking. By identifying genes in the path, what it does is it allows us to figure out which pathways are affected. So if we know this gene is affected in this pathway, then and then that leads to this protein being missing, can we do something to either put that protein back or downstream of that protein, put in something else? So it's more to direct, to identify the missing pieces so that we can develop therapeutics that are not genetics.
Okay.
So the approach is the genetics helps identify, but the approach is therapy is not genetic.
Got it. Thank you.
Hi. In one of the previous slides, it was the environmental factors you mentioned as well as genetics. Could you be a bit more specific as to what environmental factors are?
Yeah, I get that question a lot. And it's really hard to answer. But it can be prenatal exposures to certain medications. It can be trauma at birth. Essentially, it can be pretty much anything, but it's harder to study environment because it's harder to isolate a group where they've been just exposed to one thing. It's easier to study genetics. I can't comment on kind of all of the environmental factors that have been studied, but Evtiki most likely can.